Okay, today the topic of class is meiosis, so I'd like you to go to your biology Google file folder and open up Meiosis 2020. Okay, meiosis makes it so every sibling, unless you're identical twins, looks different from each other. If all of her eggs were the same and all of his sperm were the same, all of their children would look the same. But during meiosis, variety occurs because genes on his sperm are swapped and genes on her eggs are swapped, and they are able to produce unique offspring. It just depends on which egg or sperm they got. And there are so many different combinations of your thousands of genes. So we are going to get started on meiosis. Let's begin. Meiosis is a process that produces 4 egg or sperm that are half the size of their parent cell. Therefore, because they're half the size, not only is the DNA half, but the cytoplasm's also half. It's called a reduction division because it's reducing in size. They also have half the amount of chromosomes as a somatic cell. So what do I mean by a somatic cell? That's pretty simple. Look right here. A somatic cell is any non-egg or sperm. So a somatic cell has twice the amount of chromosomes, and an egg or sperm, which is called a gamete, has half the amount. Okay, so somatic cells are anything that is not an egg or sperm. So there's your gametes there, there's your egg, there's your sperm. There's a thumbprint, that's somatic, that's a body cell, that's a sperm, that's a gamete or a germ cell. So I will go here. Why does it have to be half? So let's see. That would be the karyotype or the chromosome picture of a sperm cell, and that is the chromosome picture of an egg cell. When you put the sperm cells 23 together and the egg cells 23 together, you get a baby that has all 46. Okay, so why is it important that egg and sperm each have half? So they each contribute half of the DNA to the child. Let's go back up like that. Scoot that up. Go down like that. All right, these blanks down here. Gametes, also called egg and sperm, are a new term called haploid. That sounds hard, but it just means that they have half. I like to think of haploid as halfloid. They have half the chromosomes as a regular normal body cell, like a stomach cell. They are haploid, so they have half the chromosomes in their egg or sperm, which means they have 1 chromatid per chromosome pair. They are represented usually by an N in science. Then you have body cells, which are non-sex cells. So I'm going to use the word somatic. Cells are diploid, which means they have double dip. They have 2 chromosomes per chromosome pair. They are labeled 2N. So this is a diploid cell because it has 2 chromatids per chromosome pair. So let's go here and look at a picture of diploid versus haploid. There we have a haploid cell, and here we have them doubled. Looks like you have the maternal one and the paternal one, maternal-paternal, maternal-paternal. So this would be after sex, before sex, egg or sperm, after egg and sperm combined. Let's see. Let's go to this slide next. I want to see if you understand that. Let's look at this horse. Let's look at the blanks that I would like you to fill in. So every species has a different chromosome number. Let's see if I have that on here. There we go. Look, men are man. Men and women, don't worry about those numbers. Those are just citing sources, have 46. That's our diploid number. So egg and sperm would have 23. 38 here. Each egg and sperm would have 19. Notice how these are all even numbers because they are double the haploid number. So a fruit fly in their body cells would have 8 chromosomes, whereas the sperm would have 4 and the egg would have 4. So go here and fill in the blank. Somatic cells of a horse have 64 chromosomes. That is a somatic body cell. That means that 64 is the blank, or 2N number? What would you put there? 2N is for diploid. Male gametes, sex cells, are called blank and have blank chromosomes and are called blank cells. Pausing so you can figure it out, this paragraph right here. What is the name of any male sex cell? How many chromosomes? And are they diploid or haploid? All right, and then down here, when a gamete from both parents comes together, this results in an embryo that has a complete set of blank chromosomes, which is a blank circle, or just delete the one that's wrong, cell. Pausing so you can go back and read your answers. All right, somatic cells of a horse have 64 chromosomes. That means that 64 is the, I think I'll write in caps, diploid number, or the 2N number. Male gametes or sex cells are called sperm. And they will have half of the diploid number. So that would be 32 chromosomes, and they are called haploid cells. When gametes from both parents come together, this results in an embryo that has a complete set of 64 chromosomes, which is the diploid number. Because when egg and sperm meet, they're 32 plus 32 equals the 64. All right, that is diploid versus haploid. Let's finish this page right here. The parent cells from sperm are called spermatocytes, and they're located in the testicles, and oocytes are in the ovaries in meiosis. Okay, let's erase this right here. Females are born with all of their eggs. That's around 300,000 egg cells. That does not mean a female is going to have 300,000 periods. Only 300 to 400 of those eggs will mature into a potential child. So meiosis for a female is done before they're born, when they're in their mother's womb. But males produce millions of sperm per day. So let's go here. They produce these sperm. These spermatocytes that make this sperm are located in the testicles, in these tubes right here called seminiferous tubules. And these seminiferous tubules have pre-sperm cells that do meiosis on a daily frequent basis and release them into the epididymis for storage. And then the ovaries, there's no clear picture on that. They have a similar structure where they have the pre-egg cell that did meiosis to produce the 300,000 eggs. All right, last part of this page, I want to just remember from, I believe, more like November, these basic words about chromosomes. So ignore this. This is not part one. We're deeper in the lesson than that. So this right here, when it's by itself, it's called a chromosome. It's also called the chromosome when it's replicated. I'll put that right there again. This would be an identical copy of the same gene. I'm going to go ahead and do this. Add a shape in there, please. Add a call out like this. We forgot one. Put that in the middle. That's the center of the chromosome. We're going to label this the centromere. That holds the replicated sides together. This right here will be one of the replicated sides. This is the sister chromatid. And this over here is the exact same thing. It is the sister chromatid. Okay, so two things are labeled chromosomes. You basically count chromosomes by the amount of centromeres they have. So there's a centromere, so that's one chromosome. There's a centromere right there. That's another chromosome. Okay, so what I want you guys to do is get out a separate sheet of paper. You can take it from your notebook or it can be a computer paper. Whatever you have is fine. At the end of it, you will copy and paste this picture. So take a picture, copy and paste it to the final slide in your Meiosis 1920 presentation. So it says copy and paste your handwritten Meiosis notes to this slide. So let's begin. So please label this meiosis. Now, meiosis goes through the same process as mitosis. We did the whole cell cycle where it had interphase, and then it has the PMAT for prophase, metaphase, anaphase, and telophase. But it happens twice. So you start with interphase, and then you have prophase 1, metaphase 1, anaphase 1, telophase 1, and then it does PMAT again. No interphase, though. So it's going to be prophase 2, metaphase 2, anaphase 2, telophase 2. Okay, so we're not going to look at every single one of those cycles because honestly, that's not what's important about meiosis. What's important about meiosis is why you look different from your siblings. What caused your mom's eggs to look different from each other and what caused your dad's sperm to look different from each other. That's where we're headed with this. So we're just going to look at one pair of chromosomes. So let's just see. I want to make sure I'm super clear on what we're looking at. I'm going to type in a karyotype. There we go. Click here. We are looking at one of these sets, just one of them, and it has various genes on it. We're just going to focus on 2 separate genes. Okay, so let's try to make it big. There's not going to be very much. We'll draw our cell around it in a second. So inside of this person's seminiferous tubules or inside of their ovary cells, they have these stem cells or these original cells that are going to spit out egg or sperm that have both a copy from mom and from dad. So they're actually diploid. More on that in a second. So let's say the gene locus on here will be for hair texture. So this person will have an allele for straight hair and an allele for curly hair. And then down here, this is actually a thing, there will be, sorry, my text right, it's crazy. There will be an allele for thin eyebrows. And then over here, the other allele for that will be like bushy eyebrows. So this will be the gene locus for brows. And this right here will be our gene locus for hair texture. And so because these are the same chromosomes of the exact same pair, so pair number 1, these are going to be called homologous chromosomes. So these 2 together are homologous chromosomes. Okay, that homo usually means same. You get 1 from mom and then 1 from dad. So let's go ahead and draw a cell around here. So where do these cells that do meiosis exist? 2 places and 2 places only, depending on your gender. One would be in the testes, in those seminiferous tubules, or in the ovaries. And these cells are actually, I'm going to put 2 in, they're going to be diploid. They're going to produce haploid cells, though. Okay, so our first step, let's go ahead and take an arrow and bring it down here. We are going to do DNA replication. So we're going to start right here with interphase. And this is going to be DNA replication. All right, so I'm going to replicate my chromosomes. So I have straight hair with thin eyebrows, so I'll get a replicated version of that as well. We'll have straight hair, thin eyebrows, and then I'll replicate my other option. curly hair, bushy. And the parent can have the same allele for a trait. Like they could have straight hair from mom and from dad, but this person is just going to have different alleles for both traits and bushy eyebrows. All right, okay, so they have successfully replicated. When you have homologous chromosomes that have replicated themselves, they are called a tetrad because tetrad means 1, 2, 3, 4, just like tri means 3 and di is 2 and mono is 1, tetra is 4. So this is going to be what we call a tetrad. And this is going to be replicated homologous chromosomes. So something special happens with replicated homologous chromosomes. This is going to be something called gene swap. Let me show you what I mean by that. Here we go. Going to this slide, going right here. This is actually pretty cool. So here's my tetrad. Their legs on the inside, so the middle legs are going to intertwine. Parts of the legs are going to break off and reattach to the other side. So part of that blue is going to be on the red side and vice versa. Let me show you this as an animation. Okay, so there's your tetrad. They've replicated. It was a little slow, sorry. They come together. There's your tetrad. Again. Oh, gene swap. We call this crossing over. And boom, now you have mixed up the genes. All right, so let's say this right here would be like a replicated version or different homologous chromosomes. Look, you have brown eyes and dark hair, not so smart, and a tongue roller. And the paternal side is a tongue roller, smart, blonde, blue eyes. If crossing over did exist, blue eyes would always have to be associated with blonde hair, being smart, and rolling your tongue. And brown eyes always would have to be associated with dark hair and not so smart and tongue roller. But because of crossing over, the smart gene can go to the dark-haired person, and being not so smart can go to the blue-eyed person. And tongue rolling is the same for both, so that doesn't really matter. But it just diversifies the gene pool because it swaps the genes between the chromatids. Kind of cool. Okay, so this is where we are going to have gene swapping. So this right here is going to be prophase 1, and in this phase, we're going to have crossing over, which is the same as gene swapping. And the whole purpose of this is to make egg and sperm unique from each other. So not every single egg is the same, and not every single sperm is the same. So you actually have diversity among your siblings. Okay, so let's see. Let's go down here. Let's show the product of this. Let's see. Let's do straight hair will be the same with the thin brow and straight hair, but we swap genes and let's put a bushy eyebrow with straight hair. That's a possibility now. See, that one swapped there and that one's going to swap over there. Let's see. For this one, the hair alleles we're not going to change, but they could. We got thin brows with curly hair, and we still have over here this opportunity as well. So now this is going to be your product of crossing over. Okay, so this is going to be meiosis rapid fire. Let's go ahead and just bring it back to the cell. Let's draw a circle around it. Okay, so basically, if you can visualize what I'm about to show you, then you can see any meiosis diagram and understand it. So basically, from this cell, you are going to make 4 sperm or 4 eggs. And each chromatid has to go to one of the sperm or one of the eggs. So this will go to sperm number 1. This one will go to sperm number 2. This one will go to sperm number 3. This one will go to sperm number 4, or egg just the same. So basically, meiosis 1, you cut it down the middle here. Okay, so after meiosis 1, the product of that would be the tetrad separated like that. Thin, straight hair, thin brow, bushman eyebrow. And then over here, the tetrad has successfully separated. Curly hair, thin brow, bush brow. All right, so this right here represents meiosis 1, and this is just when tetrads separate. And meiosis 2 is just an additional division. So meiosis 2 is going to separate the sister chromatids from each other. So here you separate tetrads, here you separate X's. So meiosis 2, you're going to get sperm 1, sperm 2, sperm 3, sperm 4. So let's go ahead and wrap this up. I'm going to make sperm because they're easier to see. Okay. Keep in mind, this is a considerably smaller cytoplasm because it is a reduction division. Okay, so we will put this chromatid right here. So we'll have the sperm would give the child straight hair and thin eyebrows. And this sperm would give the child straight hair and bushy eyebrows. This one would give the child curly hair and thin eyebrows. This one would give your child curly hair and bushy eyebrows. So this right here is meiosis 2, and this is where you separate sister chromatids. And just to remind you, sister chromatids is 1/2 of an X. So here I have 1, 2, 3, 4 sister chromatids. And you are resulted with 4 sperm. All right, let me see. Let me finish off with an animation. Let's go here. Cells alive. Meiosis. Okay, watch. One more time. And they separate. And you've made 4 egg or 4 sperm. All right, let's see. Down here, you have one more slide. There's one more way that you change the diversity in a gene pool, and this is so easy. So here you have chromosome pair number 1 out of 23, chromosome pair number 2 out of 23, and you get ready to split up the tetrads. Okay, so here's tetrad 1, tetrad 2. So what can happen is that these tetrads right here, these sister chromatids can flip-flop spaces. You can have this arrangement where blue goes with blue and the red goes with red, or they can flip-flop right here, and red can go with blue and blue can go with red. So they flip-flop their space. And as a result, you get different options for your sperm compared to when you had this orientation. So this right here is called, I'm going to add it in here, the law of independent assortment. And I'm not going to define that because it's defined down there. That term makes something so easy sound so hard. They line up independently of each other. Like they don't tell each other, oh, you have to be on the left and you have to be on the right. They can do it randomly. That creates, again, diversity in the gene pool. And then I forgot to label this up here. This is crossing over is a formal term, but I like gene swap better. And that is why you can have 21 kids and they can all look different from each other.